All right, so there's a model up on the board. And so anytime we look at a model, we want to anchor ourselves, figure out what is one thing I know for sure in that model. So for you, it might be the word cytoplasm, or it might be that you recognize the double helix structure of DNA. Whatever it is for you, you want to anchor yourself and then try to figure out everything else that's represented in that model. Besides the word cytoplasm, we've also got a word up here, transcription, and a word here, translation. Okay, so hopefully you figured out by now that what we're seeing is the inside of a cell. And we've got the nucleus, and we know that that's the nucleus because there's DNA molecules inside there, and we recognize that by the double helix structure, and the base pairs of A's and T's, G's and C's. And then this might be a little bit more confusing as to what's going on here. Out here we have some Golgi apparatus, and then we have a big blue blob, but there are some smaller blue circles that are attached to a tube. So hopefully you figured out that that must be the endoplasmic reticulum, and the blue circles are ribosomes attached to the ER, so that's rough ER. And just like, you know, if you're standing outside, a tree that is closer to you looks bigger than a tree far away. A ribosome that is close to you is huge, right in front of your face, and the ribosomes that are further away are smaller. So remember that we're trying to model something that is actually three-dimensional. Okay, so I'm gonna hand my phone over to my daughter. Hello, daughter.
Hi.
And she's gonna film me doing the rest of this instruction. So what I'm going to do for you here is a play. Basically, I'm gonna put on a play, and the play is called Protein Synthesis in 2 acts. And we've learned that Act 1 is transcription, which occurs in the nucleus. And then Act 2 is translation, which occurs at a ribosome out in the cytoplasm. So let's start with Act 1. So we've got all of these DNA molecules in the nucleus. One little section of one DNA molecule happens to hold the recipe or the gene for a particular protein that the cell needs to make. But as you learned from your reading, the DNA is too wide, being that it's a double helix, to get out of these pores, which are little openings in the nucleus. And it's also too important. The cell doesn't want to risk DNA getting out and being damaged. So that's why we have to use transcription and make a copy of the recipe. So the enzyme RNA polymerase will attach at the beginning of the gene, called the promoter region, and will unzip the gene, which again is just one little section of one DNA molecule in the nucleus. So now the double helix has been separated. The hydrogen bonds that were holding the base pairs together have been broken. And now the RNA polymerase will figure out which side is the strand, the template strand that it needs to read, and then it begins reading. So the first nucleotide in the gene is T for thymine. What RNA nucleotide will complement to that thymine? And you should be remembering, okay, that would have to be adenine. So the RNA polymerase would go off and find a nucleotide for RNA, that would be the A. then it would read the next one. On the gene is A for adenine. What normally would pair with A would be T, but remember we're building an RNA molecule. We're building messenger RNA, so we never have any T's, so that would have to be a U. And that would continue, that reading and pairing would continue. And so I'm just gonna fast track here until the RNA polymerase builds an entire strand, single strand, of mRNA that is complementary to the recipe on the gene. At that point, because RNA is single-stranded, it is able to fit right through a pore of the nucleus, go out into the cytoplasm, and probably hitch a ride on the ER. As that's happening, the DNA that had been separated by the RNA polymerase immediately zips right back together, and it's as if nothing ever happened, and it's completely safe and contained in the nucleus. Okay, so in that way, the gene never leaves the nucleus, it stays right there. So that's the end of Act 1, that's transcription. So now we'll move to Act 2. So here's our mRNA, messenger RNA. It's going to travel along the ER. Notice that here we have some ribosomes attached to the ER, so now it's rough ER. And eventually, the messenger RNA will insert itself into a ribosome. Now remember, we learned that a ribosome is completely made up of rRNA. And rRNA is just single-stranded RNA that happens to be all coiled up into clumps. So this is just full of rRNA. And we also are learning that a ribosome is not quite as simple as a little dot. It's actually made of two subunits, a big part and a little part, and they clamp around the messenger RNA. So now we begin with our translation. The ribosome reads the first codon in the messenger RNA. Remember, a codon is a series of 3 bases, the first 3 bases. So the first codon is AUG. The ribosome will pull in the anticodon on a tRNA molecule that complements or pairs with AUG. So here I have tRNA molecules. Remember that a tRNA molecule is just a type of RNA that has an anticodon at the bottom and a particular amino acid at the top. So can you find a tRNA molecule that has the anticodon corresponding to AUG? And hopefully you've identified that UAC would be the complementary anticodon. So this tRNA molecule would lock into place in the ribosome. All of this then kind of shifts through a little bit, sort of like a cash register tape. It'll move in a little bit, and then the ribosome is going to read the second codon, GGC. So again, we want to find, is there an anticodon that complements or pairs with GGC? And you should have figured out that the tRNA molecule with the anticodon CCG will then chemically be attracted to lock into place here. So it just so happens that only 2 tRNA molecules can fit in the ribosome at one time. Now that we have 2 amino acids next to each other, they will go through dehydration synthesis and a bond will form. We learned earlier that that's a peptide bond that will link the methionine and the glycine together. These are just the particular names of the amino acids carried by the particular anticodons. At that point, this first tRNA molecule will release its bond on that amino acid, leave the ribosome, go off into the cytoplasm, and find another amino acid to attach to. Now I'll ask you at this point, why would there be free-floating amino acids in a cell? So remember, amino acids are the monomers of protein. When we eat a protein like chicken or steak, we digest that protein down into its monomers, amino acids. Those amino acids leave the small intestine, go into the blood, and then the amino acid can leave the blood, go through a channel protein into a cell. So amino acids are in cells because of you eating protein. So back to our ribosome. This whole thing will shift, kind of like a cash register tape again, further in. We'll read the next codon, AGU. That will allow the anticodon, UCA, to come into the ribosome, lock into place. And a bond will form between the next two amino acids. This tRNA will release, go off into the cytoplasm, and find another amino acid to bond to. This whole thing shifts into the ribosome. The next codon will be read, GUC. The anticodon CAG will lock into place. A peptide bond will form between the two amino acids. The tRNA molecule will release, go off to the cytoplasm, find another amino acid to bond to. This whole thing shifts in a little bit. And this keeps happening and happening and happening until a stop codon is reached. So if you check your notes, UAA is one of the stop codons. So that's a signal to the ribosome to stop the process of translation. The protein is complete. So there is no tRNA molecule to come in to correspond to that. So our last tRNA leaves. The messenger RNA has been used. This is really just disposable. So this will all get lysed up, get chopped up into individual nucleotides that can be recycled and used another time. So now we have our series of amino acids, which makes a polypeptide or a protein. Now, this is a very simplified model. In order to be a protein, we'd have to have at least 30, 40, 50, or 100 amino acids. But this would leave the ribosome and then maybe go out to the Golgi where it would get shaped and packaged in a particular way and then sent out to do its job. Okay, so protein synthesis in two acts. First is transcription, which takes place in the nucleus, and that's how messenger RNA is made according to the gene. And the second act is translation, which takes place at the ribosome, and that's when mRNA is used to build the appropriate protein, amino acid by amino acid, using tRNA molecules.